In this presentation, we'll look at some of the components of both prokaryotic and eukaryotic cells. We'll also look at how many of these components are related through the endomembrane system. To start off, I want to look at how cells vary in their size. As you can see here, there's a huge amount of variation in the size of individual cells, ranging from nerve cells, which can be several meters long in some animals, all the way down to bacterial cells, which are about 100 nanometers across. So let's start by looking at what the limitations are on cell size. There are lower limits to how small a cell can be. If the cell is too small, it simply can't contain all of the materials and components that it needs. Notably, most bacteria are smaller than the smallest eukaryotic cells. Prokaryotes do not have large organelles that simply take up space within a eukaryotic cell. The limitations on size at the other end of the spectrum are much more complex. Cells need to transport materials from one part of the cell to other parts. This transport mainly happens by diffusion or by energy-intensive transport along cytoskeletal elements. In either case, transport large distances requires either energy or time, and so being large gives a cell a disadvantage. Another factor is that the cell needs to move materials from one side of the cell membrane to the other, either into the cell or out of the cell. We'll look in more detail at how transport across the membrane works later. For now, however, suffice it to say that the larger the surface area of the membrane is, the more material can be moved across it. Now, large cells usually have more surface area. However, they also have much greater need to transport material across the membrane because of their larger volume, and more processes occurring within such a large cell. Let's look at how this works for a simple cell that is shaped like a cube. In this small cubic cell with a length, width, and height of 1 unit has a total surface area of 6 units. This is the area of one side multiplied by 6 sides. A much larger cell, which is 5 units in length, has a much larger surface area and indeed a much larger volume. This larger volume requires that a large cell move much more material through the cell membrane. However, the surface to volume ratio of the small cell is much larger than the large cell, allowing small cells to efficiently move material through the membrane. Large cells need to move much more material, but only have a slightly larger surface area through which to do it. You may wonder about how organisms get to be big. An elephant is large because it has many more cells than a mouse, not because it has much larger cells. As you can see on the right, a large organism with many cells has a very large surface area, allowing it to efficiently transport materials through the membranes. Before we look at cellular components in more detail, I want to take a moment to look at the difference between prokaryotes and eukaryotes. This type of a diagram is called a phylogenetic tree. It shows basically a hypothesis of how different organisms are related to one another. To read it, start at the root of the tree. The root indicates the ancestor of all members of the tree. Above that root, we see that the lineage splits, with bacteria on the left and archaea and eukaryotes on the right. The tree does not show what the ancestors looked like, but we can safely assume that they were prokaryotic, since prokaryotes appear both on the left and the right side of that split. Biologists used to think that biologists and archaea were closely related because of the many characteristics that they have in common. Most notably, both bacteria and archaea are single-celled, are very small, and most importantly, have no nucleus. However, more recent genetic and biochemical analyses show that archaea and eukaryotes are much more closely related to each other than either are to bacteria. So let's look at some of the characteristics of prokaryotes. Like eukaryotes, prokaryotes need to separate the material inside the cell from the rest of the world outside. They do this through a plasma membrane, which is structurally supported by a rigid cell wall and further protected in some bacteria by a glycocalyx. Some cells have fimbriae on their surface which help them attach to other surfaces. Most importantly, a defining character of prokaryotes is their lack of membrane-bound nucleus or organelles compared to eukaryotes. Eukaryotic nuclei house the cell's DNA, but in prokaryotes, this is contained within the nucleoid, a region of the cell containing the DNA but not bound by any membrane. Okay, let's look at eukaryotes, which have a much more complex internal structure. The most obvious feature of eukaryotes is the presence of a membrane-bound nucleus. The nucleus contains the cell's DNA, where it can be kept safe from chemicals in the cytoplasm which could otherwise degrade it. The most prominent feature of the nucleus is the nucleolus. The nucleolus is the location where ribosomal RNA combines with our ribosomal proteins and is assembled into the subunits of the ribosomes. As we will learn in a minute, ribosomes are responsible for the manufacturing of proteins. They are therefore very important to cells' function and are needed in large numbers. Because they're needed in such large numbers, they have this specialized manufacturing site, the nucleolus, within the nucleus. So the nucleolus is responsible for manufacturing ribosomes. Those ribosomes, as well as messenger RNA, need to be able to get from inside the nucleus to the outside. Proteins that are manufactured outside the nucleus need to get in. They're able to travel through the nuclear envelope through large nuclear pores, large protein complexes that allow movement of other large materials through that membrane. You'll notice in this picture that the membrane itself is a double lipid bilayer. Each part of the double membrane is itself a lipid bilayer. Notice that the outer layer of the nuclear envelope is continuous with the endoplasmic reticulum, and the interior of the membrane space, called the lumen, extends through the nuclear envelope and into the ER. The phospholipids that make up the endomembrane system are manufactured primarily in the endoplasmic reticulum itself, and are then added to the membrane. When we look in more detail at the structure of the membranes, we'll see that they are very fluid, with phospholipids and other membrane components moving around extensively. There are actually two types of ER. The first is called smooth ER because it lacks the ribosomes that characterize the rough ER. Smooth ER is used as a manufacturing site for lipids and carbohydrates. It also serves as a site where drugs and toxins can be detoxified. And we see a proliferation of such smooth ER in cells that are exposed to these toxins. The rough ER is rough because of the presence of many ribosomes. Remember that ribosomes are manufactured in the nucleolus and are transported to the endoplasmic reticulum to serve as sites of protein synthesis. Although we'll spend much more time learning about it later, let's briefly review how protein synthesis happens. This is a diagram that we've seen before. Remember that the genetic code is stored in the DNA and then written onto messenger RNA or mRNA. This all happens in the nucleus where the DNA remains. The mRNA, however, can exit the nucleus through the nuclear pore. Once it exits the nucleus, it binds to a ribosome in the cytoplasm. The ribosome reads the code on the mRNA and translates the code into a polypeptide, the primary structure of a protein. Sometimes this manufacturing process happens in the cytosol, the liquid portion of the cytoplasm. Often, however, the polypeptide is manufactured on the surface of the rough ER. For proteins that need to be further processed by the endomembrane system, the ribosome needs to find its way and dock with the endoplasmic reticulum. It does this by following a signal encoded in the first few amino acids of a polypeptide. That first part of the polypeptide is called the signal sequence. That signal sequence is recognized by a signal recognition protein, which itself docks on the receptor molecules on the surface of the ER. Once it's on the surface of the ER, manufacturing of the polypeptide can continue, with the polypeptide being inserted directly into the lumen of the rough endoplasmic reticulum. When the manufacturing of the protein is complete, the ribosome, mRNA, and polypeptide all dissociate from one another. Once a polypeptide has been synthesized, it often needs to be modified. The protein needs to be folded and often have extra molecules attached, as is the case with glycoproteins, which are proteins combined with carbohydrates. This process happens in the rough ER, and once complete, the protein ends up in the transport vesicle at the outer edge of the ER. The transport vesicle buds off the main part of the ER and is transported to the back side of the Golgi apparatus. Once it arrives at the Golgi apparatus, it fuses with what's called the cis face of that Golgi apparatus. Now, some modifications continue to be made to proteins in the Golgi apparatus. However, the main function of the Golgi is as an addressing and delivery system. The Golgi apparatus sorts proteins by having clusters of similar receptors located in its membrane. Those receptors bind to tags on the proteins, causing the proteins of particular types to be clustered together in patches on the membrane. As the proteins move through the Golgi apparatus, these vesicles mature and eventually bud off into vesicles when they reach the transface of the Golgi. Each of the vesicles now contains proteins concentrated and sorted by type, and the vesicles can be taken to their destination, other organelles or the plasma membrane if the protein is being exported. You can see that here are vesicles that are pinching off the transface of the Golgi and fusing with the plasma membrane or being transported to other parts of the cell. One common fate of vesicles leaving the transface of the Golgi is that they contain digestive enzymes. These packets of digestive enzymes eventually fuse with and mature into lysosomes, which you can think of as cellular recycling centers. So, lysosomes are essentially recycling centers which contain high concentrations of digestive enzymes. Lysosomes exemplify another benefit of the endomembrane system present in eukaryotes. That is, they allow compartmentalization, which separates the inside of the lysosome from the rest of the cell. This has a number of benefits. First, the chemical reactions can happen very quickly because they can maintain optimal conditions for those specific reactions. In the case of lysosomes, the enzymes need an acidic environment, which is maintained in the lysosome. Additionally, compartmentalization allows reactants to be concentrated as the speed of reactions increases with concentration of reactants. Finally, compartmentalization allows different enzymes that function together to be held together. If a particular reaction requires 5 different enzymes, then it helps the cell to have all 5 of them concentrated and acting in the same physical location. Lastly, compartmentalization allows cells to isolate dangerous chemicals that could react with other cellular components. In the case of lysosomes, the membrane of the lysosome can fuse with a vacuole of food or even other cells that the cell has taken in from outside in a process called phagocytosis. Once fused, the enzymes complete the digestion of the food within the fused organelle. In other cases, lysosomes can digest cellular components that are worn out, such as the mitochondrion shown here. This process is termed autophagy and returns all of the macromolecules or their components to the cell for reuse. Before we move on to other components of the cell, I just wanted to review the main functions of parts of the endomembrane system with this little cartoon. You can think of the endomembrane system as a factory. Ribosomes are responsible for making a protein, which is then modified in the endoplasmic reticulum. After modification, it travels to the Golgi apparatus, where it's further modified, sorted, and packaged into transport vesicles. Those transport vesicles are marked with their destination and are transported to it. Now, one of the ways we know so much about how the endomembrane system functions in cells is through the use of pulse chase experiments. In a pulse chase experiment like this, radioactive amino acids are used to identify where in the cell particular proteins go. Before the experiment starts, cells are grown using standard media. Then, as the experiment starts, the cells are bathed in radio-labeled amino acids. This is called the pulse and is followed by the chase, in which the radio-labeled amino acids are washed away and replaced with normal amino acids. During the chase, cells are placed on photographic paper during various periods, and the radioactivity inside the cells exposes the photographic paper, indicating the regions of the cell where radio-labeled amino acids are located. This graph shows results from such an experiment. You can see that at time 0, after the cells have been bathed in the radio-labeled amino acids, these amino acids have been taken into the cell and are present in the rough endoplasmic reticulum. After several more minutes, those radio-labeled amino acids are present in high numbers in the Golgi apparatus and later in the secretory vesicles. This indicates that first, the amino acids are incorporated into molecules located within the rough ER. Then, these molecules are transported to the Golgi apparatus before finally appearing in the secretory vesicles. We can use these results to estimate how long molecules remain in the Golgi apparatus. You can see that they only start appearing in large numbers in the Golgi apparatus after about 7 minutes. However, after 37 minutes, they're starting to appear in the secretory vesicles. This indicates that some of them spend as little as about 30 minutes within the Golgi apparatus. Before leaving the endomembrane system, there are 2 more organelles of note. The first of these is the vacuole, which is present in place of lysosomes in plants, fungi, and other groups. In fact, in plants, the vacuole takes up most of the volume of the cell. Vacuoles contain some specialized reactions, but they're generally storage centers. They're particularly prominent in plants, where they play a role in storing water, as well as proteins, and sometimes noxious chemicals that make the plant unpalatable to herbivores. The next of these organelles is the peroxisome, which is the site of redox reactions. Redox reactions involve transfers of electrons to produce hydrogen peroxide as a byproduct. Now, hydrogen peroxide is highly reactive and could easily damage other cellular components. The peroxisome contains these reactions within the membrane, preventing such damage. Peroxisomes contain the enzyme catalase, which rapidly converts hydrogen peroxide into oxygen and water. Prominent and very important organelles that we haven't looked at yet are the mitochondria and chloroplasts. Plant and animal cells have mitochondria, but only plant cells have chloroplasts. Both of these organelles are involved in energy processing for the cell. Since we'll talk about them in much more detail later, suffice it to say that chloroplasts are responsible for converting light energy from the sun into chemical energy in the form of glucose. Mitochondria convert chemical energy, such as that in glucose, into ATP, which cells use as a sort of energy currency. Interestingly, biologists believe that both mitochondria and chloroplasts have a prokaryotic origin. This is the basis of the endosymbiont theory, which says that mitochondria and chloroplasts originated as bacteria and that they were engulfed into eukaryotic cells. There are a number of lines of evidence that support this. First, chloroplasts and mitochondria have a double membrane. These organelles have their own DNA that's separate from the DNA of the nucleus. In fact, this DNA is circular, as is the case with prokaryotic DNA. Third, these organelles have ribosomes, which enable them to convert the information encoded in the DNA into proteins. Lastly, these organelles reproduce and grow independently of growth and reproduction of the cell itself.